what advice would you give to researchers who are just starting in their field and face some of the same challenges like mastering complex sample preparation?
Yes. So, the basic idea of biological research is do hands on research until and unless you're handling cells yourself, until and unless you're centrifuging that lysate, you won't exactly be able to understand what you're handling. Coming from somebody like me, trust me, I was so skeptical about getting into research. I had no idea since I did not have a background in research before. But I was always interested about handling chemicals, handling samples, and working in a lab. So that has always intrigued me in some way. And that particular interest kind of got me build my portfolio in a way that yes, I can say that I can do research. Probably I can. But that does not come with failing at maybe 100 experiments every day. So it keeps happening. So I would say keep your motivation alive. Whatever you're doing is for a very, very noble purpose, very noble cause. You're trying, although you might feel yourself like a drop in a huge ocean, but that contribution that you're doing by identifying a target, by trying to work with that target so that you can achieve those specific aims for your project, for your hypothesis, is actually building a huge portfolio in the preventative medicine world, in the therapeutic world. So that's one thing to go ahead with the motivation. And sometimes, yes, you can have very, very long days in your lab where you might be doing just one experiment, but you might need to troubleshoot for another 4 hours in order to understand where you might be going wrong. But since biological samples, they are a bit tricky to handle. You're working with this very complex milieu, right, where you have things ranging from RNAs to DNAs to proteins to minerals, and everything is there. So you're handling that entire pool of things, pool of molecules. So one molecule might interfere with the other molecule, and your target protein of interest or your target molecule of interest might get degraded. Be ready for that, but at the same time exercise caution. Go through your protocols very well. Prepare as many things as you can do beforehand to get that particular result that you're trying to achieve as smoothly as you can. Again, at the same time, nothing is as smooth as it sounds. Handle your samples with care, and you will surely achieve what you're planning to. That could be like 50% of what you might have targeted before, but it still goes a long way. So do not lose that motivation and keep working.